Okay. So before we get into today's lecture, maybe I'll wax philosophical for a couple of minutes, just in response to a few emails that I've gotten regarding the grades. I posted the grades and such. One was, the comment was that the student was looking at the grades and only 25% of the class had a B or above, and everybody else had a, quote, bad grade. One is, if you look closer, it's more like 30% of the class has a B or better. So some math issues there. Also, remember, those don't include the iClicker points. So I'm preaching to the choir here, but those of you who are in here and you're in here every lecture and you're responding, we can assume that you'll have at least 5 to 7 points, something like that. So my projection is pretty close to what I've seen in past classes, that 40% of the class is going to have an A or a B, a B minus or higher by the time this is done. Also, it looks like nearly 20 students, I'll bet, look to be on track for finishing this course with a 100 or higher, which would be the highest percentage of any class I've taught in at least the last 15 years. So there are some students in here who are doing exceptionally well. Yes, you're one of them? You didn't have to raise your hand if you're in that group. No.
I'm sorry. I was going to ask, can someone donate points? Like if they have 100 points or higher, like, hey, I'll donate you.
I don't know. Can you guys sell these on Craigslist or something? I don't know. I'm just reporting the news here. Yes. I'm sorry. 10 points maximum. So let me make sure you understand how. Yes.
10 points like added onto the grade, or 10
So let's be clear. I'm literally giving you a full letter grade. So the max. Oh, somebody's going, whoa. The maximum is 10 points added to that final average. So whatever your average is now, if you got a 77, whatever points you earn get added on. I'm only saying that about 5 to 7 points because 40% of those points come from participation. So if you answered every single slide wrong, but you're here for every single one, you'd get 4 of the 10 points right there. Assume you get a couple of them correct, especially these 10-point questions at the beginning, you're probably going to get 6 or 7 points at least. So what I'm going to do is I'm going to look at not the maximum number of points that could be earned, but which student has the maximum numbers. So let's say it's 1,000. I don't know, it's somewhere around there. That student gets 10 points. If you have 900, you get 9. If you have 700, you get 7. So it's prorated down. But you're probably, by coming and participating here, you're guaranteeing yourself somewhere between probably a half to a 3/4 of a letter grade. So that means if you get 7 points and you're at an 83, that bumps you up to the 90 or the cutoff for the A minus. All right. So my projection is that's pretty much the way it's going to break out. So if you're in the C range, you're already, sort of swimming around in the 50% pond. You're sort of at the low end of you're at the shallow end of the pool at that point. Okay. The other is a C isn't a bad grade. Students come in from this course. I don't know. I'm thinking what flashed through my head was some past episode of Modern Family. Yeah, I watch TV too. Modern Family, and the one daughter was at community college, and she's having trauma in her life, as she always does, and she's not going to class, and she's not taking the exams, and she's not participating at all, so she'll probably get a C. Okay. A C, through the long storied history of academia, means satisfactory. You're doing average. You're doing about as well as everybody else, and that's where the C's are, in the middle of the bell-shaped curve. Now, a C minus, if you're a biology major, means you slid by. A C minus is accepted. I don't know why, but it is accepted towards your degree. So a bad grade is a D plus or lower, because that means you're going to have to take the course again if you're a biology major. And only about 10% of the class is in that region now. The other is, and the reason I post these grades, is the world is competitive. You guys, has anybody looked at those jobs descriptions I posted up? Do you have qualifications for any of those jobs? No. Okay. You need additional skills beyond it. So there are good paying jobs out there, but just coming to class is just the beginning of what you need to do. The other is 80% of you are in the health interested area. If you've been up to the third, they don't like me telling you this, but it's just numbers. If you go up to the third floor, the student services, they have their wall of fame. All right. And if anybody who's interested in pre-med seen the pictures of the students who got admitted to one of the professional schools. And it's about 75 or 80 a year. It's been pretty consistent. The frame of pictures isn't getting bigger, right? It's about the same size all the way across. Well, by my rough estimate, about 40% of all the students who are going to take genetics this year are in this class right now. So, and if it's about 80, that means statistically about 32 students are going to get admitted out of this class. The students who currently have an A in the course are more than enough to account for all those 32. So it's just, and that's why I post your grades as it is. It's a competitive system out there. Anybody know what I'm talking about? The sort of now somewhat infamous "You're Not Special" commencement address. If you haven't heard it, listen to it. Okay, regardless of what your parents are saying, my daughter is the only one who's special. Okay. The rest of the world views you as adults, and especially in a lot of these competitive programs, they're not going to see that you had some personal issue or something else. That won't come up until you're on that short list. So you need to be doing everything you can to be gaining additional experience and setting yourself apart from the rest of the crowd. Okay. So as far as the grades go, when I post the fourth grade, the grades after the fourth exam, all the iClicker points will be included at that point. So when you need to make the decision of whether you're going to take the final exam or not, you will have your final course grade. If you do not take the final exam, that's what your grade will be. Okay? Any questions before I get off my soapbox and get back to what I'm supposed to be doing here? Okay. All right, so we're in this chapter 18 on DNA repair. It's really got 3 sections to it. It's a long chapter. We're really going to be covering the first section. We're going to finish the first section, which is on the types of DNA mutations that can happen. The second section is on how those mutations can happen. And the third section, which we will attack next Monday, because everybody probably knows Wednesdays. Yeah, that didn't get past you, right? Okay. All right. So, yeah, Dr. Hartman wanted me to do a guest lecture on Wednesday, and it's like, I'm fine with that, but, speaking to an empty room is a little demoralizing. Okay, so on next Monday, we'll be looking at the third section, which is how the cells repair that damage. So before we get started on that, let's try our 2 questions. So this is where we ended last time, and I'm going to start here again today. But let's see how much of it. No, I'm sorry, not that question. This goes back a little further. It's not where we ended. I changed my mind at the last minute. Okay, let's go to 1 minute on this one. Okay. So again, make sure you always put some kind of response in, at least get the participation points. Okay, last few seconds. All right. Does the question look familiar? Yes, no? Okay. It kinda? Yeah, that's it. Take the middle of the road. Okay. Same question I gave you last time. I just changed the answers around a little bit. All right. So this goes actually from the previous chapter. Any questions on that? Okay. Now, from the chapter we're going to do today, This one you should be able to answer even if you didn't look at the notes. Should be pretty much a giveaway. Oh, look, 93 getting that. Okay. If you make the question simple enough, everybody can answer it. Except for a few lost, lonely souls. Yeah, we'll go to 1 minute on this. All right. All right. So what's the answer? So which one of these isn't a DNA damaging agent? Oh, I should ask that question. They all are. We're going to talk about that. Okay. So as I said, this is chapter 18. This is on genetic mutations, DNA repair. Again, it's probably an area that hasn't been covered very well in other courses, so a lot of this may be a little bit new. Or a lot new, yes? Yes. I think so. We should be pretty close now. It might be off by half a lecture. Some of the other chapters are a little bit shorter. This is the longest chapter. Okay. So we ended with this idea of intragenic and intergenic suppression. And I know the intergenic suppression can be a little bit confusing. So it seemed like a good place to stop and now a good place to start again. So this is close to where we ended last time. So we're talking about suppressor mutations. Suppressor mutations mean a type of mutation that actually brings you back to a wild type phenotype. So you're suppressing the particular mutant phenotype that you're looking at. So here we have a point mutation. We looked at this before. So you have a leucine codon. We have a mutation that changes the third codon. So now it codes for phenylalanine. So would this be a forward or reverse mutation? This is what we talked about just before this. Okay, does this look like the typical kind of genetic mutations that Mendel and everyone else were probably looking at? You have a wild type mutation, you get some mutation, you get some mutant phenotype. So this is a forward mutation, which is your traditional type of mutation. Now, if there was a second mutation that changed this nucleotide back to a T, this would be a reverse mutation. You're going back to the original wild type DNA sequence. Okay? That's not a suppressor mutation. This would be a reverse mutation. The suppressor mutation is a second mutation. So now you've actually changed two bases in the codon, but because the genetic code is degenerate, you've actually changed it back to a leucine. So you're back to the wild type phenotype, but it's two consecutive mutations within the same gene. So this is intragenic suppression. Okay. Yes? No, this would not. The reverse mutation would be this A being mutated back to a T. So that now it was AAT again. So it's back to the wild type sequence. That's a reverse mutation. Here we've had two consecutive mutations, and now we have GAA, but that also codes for leucine. So you've got two changes in the codon, but it brings you back to a wild type mutation. So this, going from here to here, is a suppressor mutation. There's a question? Yeah.
And just to be completely sure, it must be grouping at repair.
No, it's just a way. You could have another mutation. This is the most likely way that it would happen, but it could be any mutation within that gene that brings you back to a wild type phenotype. So you're suppressing the mutant phenotype and getting back to wild type. And it's intragenic because the mutations are within the same gene. Okay? Yes.
So then, in order for it to be a reverse mutation, it has to go back to the
the original.
sequence.
Right.
Okay.
So the mutation is basically getting reversed to what it originally was in the wild type sequence. Right. The outcome is the same. It's just molecularly how it happened. If you sequence this one, if you had a reverse mutation and you sequence the DNA, you'd never even know the mutation was there. If you sequence this DNA, you would see that it's been changed, but you would understand why you're getting a wild type phenotype again. So anytime you're going from a mutant phenotype back to a wild type, it's a suppressor mutation. Okay. Any more on this one? The intergenic is a little harder, I think, to conceptualize, but still pretty straightforward. It just means we're now going to involve two different genes. So the mutations are going to occur in two different genes. The two mutated genes are going to work together to bring you back to a wild type phenotype. The most common type of intergenic suppression is what's shown here, involves mutation of a tRNA. So, which one I can actually read. So here's the site. So you change this nucleate, you have let's see how they're doing this. So here's the mutation. Oh, okay, I see how they're doing. Okay. So we've had the first mutation would be just like we just did. All right. So let's say you had this mutation. All right, and now codes for phenylalanine. The other way to suppress it is the tRNA. Normally, the tRNA would be bringing in a phenylalanine. But if you mutated the anticodon, so it now recognized if a leucine tRNA had a mutation in the anticodon that now recognized a phenylalanine codon, then it would bring leucine into there and restore the normal sequence. So it's a mutation in a tRNA that now recognizes a different codon and brings in the amino acid. The most common type of suppressor mutations is what they're showing here. You have a nonsense mutation, so you get a very clear phenotype because you have premature termination of the polypeptide. You get a non-functional protein most of the time, and you have a mutant phenotype. But then you have a mutation in one of the tRNAs that changes the anticodon so it recognizes a stop codon. Does the cell normally have tRNAs that recognize stop codons? Please say no. Okay, remember, these are protein factors. But if you have a modified tRNA that's been mutated, so the anticodon recognizes the stop codon and brings in an amino acid, you prevent the premature termination, and in most cases you can get the rest of the protein made and restore functionality. So it's intergenic because it's mutations in two different genes that are now working together to restore the wild type phenotype. Makes sense, or you got to let that process a little bit. Oh, I can't even get anybody to nod today. Wow. Okay. All right. So, oh wow, I can write lots of questions, can't I? So here's a summary of the molecular basis and phenotypic outcome of 20 types of mutations. If you just choose to memorize the table, that's up to you. Believe it or not, we actually have covered all of these. Okay. And many of them are they're all interrelated. In other words, you've got the missense mutation, nonsense silent mutation, neutral mutation. Those are all the result of single base changes in a codon. It's just what the outcome is. Okay. All right, so let's try this question while you think about that for a minute. Oh, we'll go 1 minute on this one. Okay. So what's the answer? That's what we were just talking about. This is intergenic because you got it's mutations in 2 different genes. A mutation in gene X. Ah, that reading is. Damn English. Gotta read the question. Okay. Reading counts. Reading is your friend. Okay. At least a little material now that's probably a little easier to understand. Pretty straightforward. Looking at mutation rates, all we mean is how often does a particular gene undergo a mutation. And we can express it in several different ways, things like mutations per cell division, per gamete, or per round of replication. But in general, most of the time, it's just kind of inferred as probably per cell division. Okay. So there's this a mutation that it's a form of dwarfism. If you look at this particular gene, it's due to a mutation in this gene that occurs about 4 out of 100,000 gametes. So the frequency with which this gene is mutated is 4 times 10 to the minus 5th, or 4 out of 100,000 gametes. So we just put that in scientific notation. Okay. So the next slide is going to look a little confusing because it's going to sound very similar. So make sure you have this clearly in mind. This is just how often does this particular gene undergo a mutation per meiosis. So out of 100,000 gametes, we would expect to find this gene mutated in about 4 of them. That's the rate of mutation. Yes. You have a question? Right. So let's just think of it that way. Out of 100,000 gametes, we would expect to find this gene mutated, a mutation in 4 of those 100,000 gametes in this gene. This is the mutation, the rate of mutation. The frequency is how often does this gene, this mutation occur within a group of individuals. So from population genetics, this would also be called the allele frequency. What is the frequency of the allele? So with this same gene, if you look in the US, what you find is about 1 in 20,000 individuals have a mutation in this gene. So certain genes, for instance, we've talked about this previously with sickle cell anemia, in areas where malaria is endemic, the frequency of the allele for the sickle cell hemoglobin is higher because it actually has an increased survival value to it. So this frequency with which this gene has this mutation can vary from one population to another. So you see the difference between the rate of mutation and the allele frequency. Okay. So there's really 3 main factors that are going to affect these mutation rates. One is the obvious, in other words, what's the primary cause of the mutation? Whether it's spontaneous or it's caused by chemicals or something else that we put out into the environment, whatever is causing it. So you have the frequency with which it gets damaged, offset by the probability that it will be repaired. Next time when we talk about the DNA repair systems, they are very efficient, and so that the actual level of DNA mutations is much higher than you would expect, but these repair systems are very efficient. They're not perfect, but they're highly efficient at making those repairs. So it's really the balance of how often is it mutated versus how often is it repaired, and from the experimental or investigator side, what's the probability that we'll actually see the mutation, that we'll pick it up in our assay. So again, these frequencies that we look at are based on what the experimental system is capable of detecting. Okay. So just some general ballpark numbers. We've talked about this before. The spontaneous frequency mutation in bacteria is extremely low. In general, any given gene would be mutated in 1 out of 10 to the 8th or 10 to the 9th cells. It's higher in eukaryotes. Actually, it's several orders of magnitude higher. It's not really clear why. But the other is that the mutation frequencies and these rate of mutations vary tremendously between different species and also between different genes. All right. And why they do this isn't really well understood. Obviously, it seems that there's certain parts of the genome are more susceptible to damage, or maybe certain parts of the genome aren't repaired as well. But just like we had hotspots for recombination, there are hotspots for genetic mutations. And it varies over a pretty wide range. If we look at the causes of mutations, in two very, very broad general categories, one is spontaneous mutations. Those are just caused by natural agents, natural changes. So as you're walking to class today, you're exposed probably to enough ultraviolet light to generate some reasonable number of thymine dimers on your way over here, but your repair systems hopefully are taking care of that at this time. Those are natural consequences. There's low-level radiation naturally in the environment, cosmic radiation, some of this. The others are just the compounds that we've put out into the environment ourselves. But all of those and just sort of naturally occurring we'll put in the category of spontaneous. The induced mutations, I just generally tend to think about, these are the ones we've done deliberately under experimental conditions in the laboratory. So when Drosophila was, when early investigators were putting Drosophila into X-ray machines to create chromosome rearrangements and such, these would be induced mutations. A lot of those we were talking about auxotrophs and with E. coli and such. To get those mutants, they were generally started with a large population of cells and then treated them with some type of a mutagen that would cause these mutations. So the induced ones I'll just put in the category of the ones done deliberately in the laboratory for research purposes. Okay, yes.
So in this slide, you wouldn't move that up to spontaneous where it says reduce environmental and chemical radiation?
This one I kind of put in that iffy category. This is one of those things that I put it in the iffy category. I think you make an argument. Again, we try to make these two nice, clean areas. But I guess you could argue if it's something that we threw, if you happen to live like next door to Chernobyl, then, it is spontaneous mutations, but they were also caused by man-made events. But generally, so that's why I said induced ones. I'm not going to try to make a real clean distinction on this. I think the distinction's a little bit fuzzy, unless you're talking about clearly natural environmental issues such as radiation, ultraviolet light versus deliberately applied mutagens in the lab. Okay. So this next section talks a little bit about how these mutations come about, the basic molecular mechanisms and some of the chemistry behind it. The first category we can call are spontaneous replication errors. This goes back to when we were talking about DNA replication, the DNA polymerase. If you remember, the polymerases had a 3' to 5' exonuclease activity. Everybody remembers that, right? Yeah, just shake your head, yes, okay. This is a proofreading activity. So the idea is normally the base pairing is very, very accurate. If there's a T on the DNA template, then the DNA polymerase puts an A opposite the T. But sometimes that mispairing, there is a mispairing that happens. I'll show you a couple of examples. And that's what allows the polymerase to now remove that base and replace it with the correct base. That system is not 100% efficient. It's probably about 99% efficient. So we can get some DNA mutations that are the result of replication errors. All right. So as I said, in E. coli, this replication is very precise, making a mistake only about once out of every billion nucleotides. The main reason for many years that was thought to be the main source of this mispairing was something called tautomeric shifts. I'm not going to go into this in any detail. Basically, it was an alternate position of the electrons within the bases, which allowed the bases to make a different hydrogen bonding configuration. It looks more likely that this mispairing now is the result of the same kind of wobble effect that we talked about with the tRNAs being able to hydrogen bond with more than one base. But regardless of that source, you can end up with this mispairing. So sometimes you get T pairing with a G. Is that normal? Come on, yeah, everybody knows that. Or a C pairing with an A. So here's one of the examples. These are the so-called tautomeric shifts. The main thing to focus on is showing you how you can form two base pairs between A and C and three stable base pairs between G and T. Now this is very rare. It's going to be a very small amount of the time, but sometimes when the electrons are in the right configuration, this is how the polymerase makes a mistake. And if it doesn't recognize the mispairing, then that mispairing stays in place as the polymerase moves on. Okay, and you can get a similar wobble interaction that now allows T to base pair with G and C to base pair with A. All right. Here's an important point. You have this mispairing. Because DNA replication is semi-conservative, if that mispairing is not repaired before the next round of replication, then all opportunities to repair that mistake are gone. Does that make sense? All right, so I'm going to show you how that works. So the cell must repair this mismatch before the next round of replication, or it will become a permanent change. So here's how it happens. And it's directly a result of semi-conservative replication, the fact that the DNA strands have to separate. So here's a wild-type sequence. Trying to see. Okay. So the strands separate. We have replication. And now instead of getting the normal, so the template should be, let's see, it's base pairing with this T. So normally it should be putting in an A, but it puts in a G. This one replicates normally. If this mismatch isn't repaired, when you go to the next round of replication, then the strands separate, the cell replicates it, puts in the correct base. Now everything is completely base paired correctly, but this strand is going, this strand is AGGC, and this one's AAGC. So since that mismatch wasn't repaired prior to replication, now that DNA sequence change is permanent. So that's what we're going to see in so many of these cases when we get to how the cell repairs the DNA damage. It has to do it before the next round of replication or it becomes permanent. Okay. The other type of mutations that actually occur more often than we initially thought they did was how do you go about getting insertions and deletions? So this would be the frame shift mutations we've talked about, would be the major cause of this. There's two mechanisms that are really thought to do this. One is called strand slippage. I'll show you how this is during DNA replication. The polymerase, again, the polymerase makes a mistake. And the other is unequal crossing over. And this goes back to genetic recombination. And here you'll see that there isn't an equivalent transfer. The reciprocal exchange of the DNA during the recombination is not equal. Okay, I'll show you the examples here. All right, so this is the idea behind strand slippage, insertions and deletions. One is you're going to see these type of mutations when you have a series of the same nucleotides. That's when it happens most often. So for instance here, we have this series of T's. We've got 1, 2, 3, 4, what is it? 3, looks like 5 T's in a row. So the polymerase is going along and it's adding the same nucleotide. And in some way or another, and the mechanism really isn't clear, the polymerase kind of slips a little bit and either adds an extra base or misses a base. And so the result is, when we come out of this, so if we do it here, the polymerase is slipping a little bit, ends up adding in an extra base, and now we end up with, instead of 5 A's on the opposite strand, we end up with 6 A's. And again, if that's not fixed before the next round of replication, then we're going to end up with 6 T's on the other side. Or, because of this series of the same nucleotides, one of them loops out a little bit, and the polymerase skips it, then you end up losing a base. So these insertions and deletions are most often occurring where you have a long series of the same nucleotides in a row. And this tends to be error-prone for the polymerase. The other way this happens is so-called unequal crossing over. So again, we go back to genetic recombination. The chromosomes are supposed to be aligned base by base over their entire length. They recombine, and equivalent amounts of DNA are exchanged. But if you have repetitive sequences that are very close to each other, sometimes the DNA doesn't align exactly in the exact right place. And so in this case, this blue sequence should be shifted over several bases, but the first sequence is, but because we have these repetitive sequences, they're misaligned a little bit. So now when you come down from the red to the blue and you come down through the unequal crossing over, one chromosome has now gained DNA sequences and the other has lost it. So again, another mechanism for insertions and deletions. Okay. Kind of waiting for something to happen here.
It's a weird chapter.
It's a weird chapter. Hey, it's molecular biology. It's molecular genetics. Okay. Now let's do some chemistry. That's always fun. Oh, chemistry. Better living through chemicals. Okay. Can also cause some DNA mutations through chemical changes. We're not going to have a lot of these, but these are some very well studied examples. One is a depurination reaction. It's exactly what it says. It's the loss of a purine. What happens is we have the purine right here where I have the arrow. Remember, this is the glycosidic bond that joins the base to the 1' carbon. And this is a somewhat chemically susceptible bond. And so this bond will break sometimes. We lose the base, and we create an apurinic site. It means the purine base is missing. The sugar phosphate backbone is still in place. We just have a hole in the DNA where this purine base used to be. So in the jargon of the business, it's called an AP site for an apurinic site. You can also have apyrimidinic sites, but they're much rarer. Apurinic sites are the most common. Oh, forgot I did that. Okay. Now what happens is, if that apurinic site's not repaired before DNA replication happens, then the polymerase comes up to that missing base. There's nothing for it to pair with. And in most cases, it'll just add an incorrect nucleotide. It just takes a while to guess at it and tries to fill in and moves on from there. Okay. So what's the probability that it'll get it right? So you guys are better at math than you thought. Okay. All right. Now, does this happen very often? Well, at least if you look at mammalian cells in culture, they typically lose about 10,000 purines a day per cell. So while you're sitting in here until for the next hour, you can begin to calculate how many purine bases are being lost. It does give you an idea of how efficient the repair systems have to be. Could cells tolerate this for very long? I wouldn't think so. Okay. So here's in diagrammatic form what's going on. So we have the apurinic site right here. The base is missing. So that when you go to DNA replication, the strand is still intact. The phosphodiester bonds are still there. So the strand doesn't fall apart. But when the polymerase comes by, in this case, it just inserts an A, just to fill it in, and it continues along. What should it have inserted? It's just a test of your eyesight. Okay, it looks like, from what I'm seeing, there was a C, so we lost a G here. And so now we have an A instead of what should have been a C. So now we have this sequence, and it comes back over. And since this wasn't repaired, you're going to have the same mistake occurring again. Okay? So this is why it's so critical that these mistakes get corrected before the next round of replication, or they just get perpetuated. Okay. Another common one is a deamination reaction. That just means the removal of an amine group or an amino group. You can do this happens spontaneously at a reasonable frequency, or you can also do it with some chemicals in the lab. So, for instance, if you deaminate cytosine, you have uracil. So you have C being converted to uracil. Would you normally find uracil in DNA? No, in fact, uracil in DNA is highly mutagenic. And on Monday, we'll talk about a mechanism for specifically finding uracils in DNA and repairing them. Okay, so now the next time around, the uracil is going to base pair with what? What would it normally base pair with? With adenine. Okay, that's that reading thing again. So this is a CG to TA transition mutation. So we talked about transition mutations are like for like, transversions are like pyrimidine. So the transitions are purine for purine, pyrimidine for pyrimidine. So what was a CG base pair is now a TA base pair. So this is the transition mutations are the most common type. Okay, so this is just showing it to you in a diagrammatic form. So here's the amino group. It gets deaminated and is biochemically converted to uracil. Okay. So what's the relationship of uracil to thymine? It differs by the presence of that methyl group. Okay, so there's a methyl group on the uracil that gives you thymine. All right. And the most common type of methylation in eukaryotic cells is 5-methyl-C. Right? So what happens is you have these 5-methyl-C groups in eukaryotic DNA. They are hotspots for DNA damage because if you deaminate the 5-methyl-cytosine, you form 5-methyl-uracil, which is thymine. So you're changing a C to a T, and yet that's a completely legitimate base in the DNA, so the repair system doesn't see that as a mistake. So sequences that have high levels of methylation of the C residues tend to be hotspots for these types of mutations. Okay, so again, you get a CG to TA transition mutation. So that's what it's showing you here. Same thing we had before, except now it's 5-methyl-cytosine. Same deamination reaction. So now you have 5-methyl-uracil, which is thymine. All right, moving along. Can also use base analogs. This simply means these are chemicals that look very much like the nucleotide precursors. They can be incorporated by the polymerase into DNA, but they are analogs, they are chemically modified, and they have some different outcomes. A lot of the earlier generations of antiviral and anti-cancer agents were these type of analogs that would get incorporated into the rapidly proliferating cells. They get incorporated into the DNA, and then, as I'll show you, some of these analogs lead to high levels of mutations. And then the whole idea was to selectively kill the rapidly dividing cells. One of the most commonly used, it's been studied for years, is 5-bromouracil. It's oftentimes called 5-BU. It's thymine. It looks just like thymine, except where that methyl group is at position 5, there's a bromine. What happens is that tautomeric shift I talked about earlier, without the methyl group there, it tends to shift into the unfavorable configuration more often than it should. And so what happens is it causes it to 5-bromouracil to base pair with guanine. So the 5-bromouracil is an analog for what? That's the thymine. Right? So normally it should be a T and A pairing with the T, but now because of these tautomeric shifts, you're getting this, what is the equivalent of a T base pairing to G. So the result is a TA to CG transition mutation. And so this occurs at higher frequency in the presence of this analog because it spends more time in this unfavorable electron configuration, so there's higher levels of misincorporation by the bases. So here's the normal base, and as I said, so this is the only point that differs. It looks the same as far as the polymerases go. It's the same. It looks like the regular nucleotide, and it incorporates it into the DNA very efficiently. So so let's say you want to use this 5-bromouracil as an anti-cancer drug because the idea is that the cancer cells are rapidly proliferating. The other cells in your patient are pretty much in a homeostatic situation, very low levels of replication. If you add the 5-bromouracil, would this be toxic or would this cause DNA damage if the cells are not actively undergoing replication? Just the presence of the analog. No. You only see the increased mutation after it's been incorporated into the DNA. So that's why it was used to selectively kill rapidly proliferating cells. But you do have some side effects because there are different cell populations in your body which are naturally proliferating anyway. So this is generally why you got the side effects from a lot of these base analog anti-cancer drugs. Okay. So here would be the normal pairing of the 5-bromouracil, which is basically T base paired to adenine. But you get this tautomeric shift and this mispairing that allows the T to base pair with the G. Or this T analog. Yes?
Something like the global replacement, is it difficult to make sure that the agents get into the DNA or interact with the DNA or not?
Well, actually, the more challenging part is usually getting the analog into the cells. Once the analog is, if you have a good delivery system, and once it's incorporated, it's fairly well transferred across the cell membrane. So, but any of these kind of drugs, delivering it to the target tissue is one of the big issues that have to be solved. Yes.
So it's not a mispairing if bromouracil is with adenine, only if it's with the G because
Right. So this actually happens most of the time, but let's say this mispairing occurs 1 in 1,000 times. With this analog, this is more likely to occur probably at just make a guess, at least 10 times that level. So it's an analog that's causing this mispairing to occur at a much higher rate than would normally happen. So you're doing this, would this be induced mutations? Yes, you're deliberately manipulating the system, trying to create these mutations.
But it's still cause and effect, though, base pairing with A, even though it's not a T.
Oh yeah, no, it can be. It probably most of the time it'll base pair fine with the A. It just mispairs with G at a higher frequency than thymine would normally happen. Okay. Another group of really potent mutagens are so-called alkylating agents. These are agents that add methyl and ethyl groups to the DNA, and these modifications onto the DNA actually then change the base pairing rules. So that's how they create mutations. One really nasty one is called ethylmethane sulfonate, or EMS. While I was in the lab in California, it was a fly lab. One group of researchers were using EMS to induce mutations in the flies. So was this something you should handle very carefully? All right. What's the safest way to handle EMS? Let someone else do it. Okay. Preferably they're doing it on the weekend. All right. I joke around, but if you're in labs where they're using any of these type of compounds, please read the warnings. These are some nasty drugs or nasty compounds. So what happens is these alkylations, these ethyl and methyl groups that get added onto the bases, they interfere with the base pairing, and again, you end up with the same kind of mutations we've been looking at, where you can get these CG to TA, and in this case, it can go back and forth, TA to CG transition mutations. But they're all the result of inducing changes in the bases that then interfere with the proper base pairing during DNA synthesis. The other is deamination reactions. Have we talked about these before?
Yeah.
Good. At least we're doing okay. We're getting better. You can induce them. The deamination reactions I was talking about before, a lot of times they actually occur spontaneously, and your repair systems are quite good at fixing those. But you can also do it deliberately with nitrous acid, for instance. You can deaminate the cytosine, as we talked about before, gives you the uracil, and that's going to then base pair with adenine as it normally would. So CG to TA transition mutation. Are you seeing a theme here? Okay. But nitrous acid also deaminates guanine to form something called xanthine, which is actually a precursor to the thymine. All right. And so that gives you CG, also gives you CG to TA transitions. It also can deaminate adenine to form hypoxanthine, another precursor to these nucleotides. And that can all then base pair with the cytosine. So now you get TA to CG mutations. All right. So here we're going CA to TA, and here we're going in both directions. So what it does, it gives you the interesting possibility for me to write questions on the exam that combine the results of treating DNA with nitrous acid and forward and reverse mutations. So you can use it to get, for instance, a TA to CG transition mutation first, and then you can get a second mutation that can actually reverse it. Okay. So some classes of mutations can be reversed by additional treatment with the nitrous acid. Yes.
So how is that different from EMS?
Yeah, actually you could do the same thing with that too. Not as easy to control. Okay. All right. So here's just a summary of these different types of mutations. Here's EMS, letting you don't have to know what these intermediates are. The whole idea is that you just can see by looking at these different types of modifications to the DNA. The general mechanism is they're interfering with the proper base pairing and lead to these base substitutions. Okay. Okay. Another area that's critically important, because it's the basis of my research and hopefully my funding, is oxidative reactions. So we're aerobic organisms. Aerobic respiration is very efficient, does an excellent job of extracting energy from compounds such as glucose and such. Everybody's familiar with the electron transport system within the mitochondria. They've beaten that into you. Okay. The problem is, okay, as those electrons are kind of rolling down that potential energy hill and ultimately delivered to oxygen at the end to form water, if those reactions were 100% perfect, we really may not even need this slide. But about 2% of those reactions fall off the tracks and end up generating these oxygen derivatives, superoxide anion, for instance, peroxides. There's also some nitrogen derivatives and such. These are highly reactive oxygen species that are looking for an electron to fill out that last shell, and they're going to go steal it from anywhere they can, and so they can cause oxidative damage to major molecules in the cell, including protein and DNA and lipids and such. So that's what we mean collectively by reactive oxygen species. There's just this collection of oxygen derivatives which are highly reactive and highly damaging within the cell. You have a large number of enzymes to protect yourself from this. So for instance, the superoxide anion is probably one of the most common. So there's a key enzyme called SOD, superoxide dismutase, which breaks this down to catalase and water, and eventually, or catalase breaks this down to peroxide, and then catalase breaks it down further to water. You need these repair systems. If you look at mutants where the system has been inactivated, whether you do, let's say you knock out the SOD gene in a mouse or in flies, you end up with very severe, if not lethal, consequences. So it's critically important that the cell be able to control this level of oxidative damage within the cells. Another category of so-called really nasty compounds are intercalating agents. The key here is these are flat planar molecules. What they do is they slip in between the bases, they distort the helix, and then cause the polymerase to make mistakes during replication. All right. So these intercalating agents are common sources of another way of getting that kind of replication slippage that we talked about a little earlier. This just makes the problem even worse. So these are really potent mutagens. Here's a couple of them, proflavin and acridine orange. Also, there's a compound called aflatoxin. You may have heard about it. It's from a mold that tends to grow on peanuts that are in peanut silos and such. And it still remains, it's been known for decades and still remains one of the most potent carcinogens that we've yet identified. So don't eat peanuts that have been in the silo too long. Okay. All right. So now we'll take a break here and let you breathe. Let's try this. Yeah, it looks like 1 minute should be sufficient. Okay, well, I guess I'm going to a minute and a half. Okay, what's the answer? Well, let's see. What? Do what? An intercalating agent? That would be what we just talked about. Intercalating means it slips in between the bases. So these are flat planar molecules. They slip in between the bases and they distort the helix. And when they do, then the polymerase, as it's replicating, tends to either add or delete a base. So to go to this question, so why is it frame shift mutations? Because you could reverse it. So the mutation may have been the result of an insertion, and the intercalating agent can now lead to a deletion and restore the mutation. Okay. Could this this mutation produced by this newly developed chemical, could this be an alkylating agent? Is it likely to be an alkylating agent? Do alkylating agents cause, can they be reversed by an insertion or deletion? Yes? Probably not, because they're only changing a single base. But the alkylating agent, this is, so it says mutations produced by newly developed chemicals. So you got some chemical, this was actually the basis of something that was called the Ames assay. This was required by the federal government. It was an assay to look at different categories of DNA damaging agents. And the idea was if you could reverse the reaction with a particular type of agent, then that's probably the type of mutagen it was. So what does all that mean? If, let's say, your newly developed chemical was an alkylating agent, it's going to change it's going to cause base mutations, but probably not insertions or deletions. So now treating it with an intercalating agent that causes insertions and deletions is probably not going to restore the wild type. So that's the rationale behind the question. Okay. You didn't know there were so many ways to damage your DNA, did you? And you do it every day. All right. Now, this one's radiation. So, especially ionizing radiation like X-rays, highly damaging to DNA. They tend to break the chromosomes. So, as said here, Mueller first showed this mutagenic effect way back in 1927. And I think this is true, that somewhere maybe through the 40s or the 50s, when people didn't realize just how dangerous X-rays were, that there were little X-ray machines in shoe stores so that you could put your shoe on and kind of X-ray your foot and see how your foot was fitting in the shoe and such. That's what they say. Okay, I think it's true until it was recognized that just how damaging ionizing radiation is. So they tend to produce free radicals. Free radicals are in that category of the ROS we just talked about, so they can be damaging that way. But probably their most common trait is they make these double-stranded DNA breaks. So they tend to break the chromosomes. These broken chromosomes are very unstable. They just knit, the cell doesn't want to have these broken ends, so they fuse them together. So let's say you have two breaks in the same chromosome, so now you have three pieces, and the little piece gets lost and the two big pieces knit together. What have you just caused? You just made a deletion mutation. So you've lost a part of the chromosome. So especially these chromosome mutations in Drosophila, many of those were generated by so-called chromosome mechanics years ago by putting the flies in X-ray machines and then seeing what type of mutations came out. So the key here is these high-energy ionizing radiation, X-rays being the most common one. Cosmic radiation would fall in the same category, create these double-stranded breaks in DNA. Okay. The one that you probably know the most about is damage from ultraviolet light, and it's probably the one that your cells are most efficient at repairing. So the issue here is, in this case, this is a direct absorption of the energy. So the key is that those conjugated double bonds in the purines and the pyrimidines absorb light energy very efficiently in the ultraviolet range. So at about 254 nanometers, which is in the ultraviolet range, DNA and RNA absorb that energy. And what happens is adjacent pyrimidines form these so-called pyrimidine dimers. So this is actually a covalent bonding between two adjacent pyrimidines. And we see the same thing as we had before. It distorts the helix. When the polymerase comes by to replicate, it ends up misincorporating the base, or actually in most cases, the polymerase will stall. It can't get past these pyrimidine dimers. It tends to come off of the DNA and reinitiate downstream, and then you have a gap in the DNA during replication. Is that a problem? What's going to happen at the next round of replication? It's all going to fall apart. Okay. So we'll talk about that when we get to the mechanisms of how this is repaired. So the most common one that you probably already are familiar with are these thymine dimers, that is adjacent T residues, get this, form this covalent bonding, the 2,4,6 cyclohexane dimer. But they're also the most easily repaired. When we get to how we repair the DNA, every example I give you except this one, the idea is identify the damaged DNA, repair the damage, and replace it with new DNA. In this case, the cells do it very efficiently by just simply reversing the chemical reaction, but we'll go into that later on. But you can also get CC and CT dimers. So the TT dimers are the most common, but you can get the dimers between any two adjacent pyrimidines. Okay. So as I said, that pyrimidine dimer distorts the helix and causes the polymerase to stall. Yes?
Why does that affect only
Because it's basically the chemical structure. It facilitates a covalent bond. There's actually two covalent bonds that form between the two bases. All right. So what happens is it blocks the polymerase from replicating. It can't replicate past these pyrimidine dimers. It's one reason that UV is a very effective sterilizing agent. All right. We're talking about bacteria, not. Cell can survive if the problem is the cell has to overcome this block, and later on we'll talk about how the cells do this, but it's something called error-prone replication. So the cells are going to have a trade-off. They can either die or increase the probability that there's going to be a DNA mutation, but they might survive to the next round. But we'll get more into that on next Monday. To do this repair is something called the SOS system. So I know people haven't. How many of you know Morse code? Some of you should. That's interesting. I don't know it either, but I thought it'd be somebody who just too much time on their hands. Okay, so SOS is the old Morse code called distress signal. So this SOS repair system from E. coli gets its name for that. This is a last-ditch effort on the part of the cell to survive. So this system is only going to be activated when there's been very severe damage to the DNA. One of the most damaging, probably one of the most lethal events for the cell is stretches of single-stranded DNA. So what's going to happen is, if that, as I said before, if that polymerase gets blocked at the thymine dimers, then it disengages, re-engages, and leaves a gap in the DNA. And that's what's going to be lethal if it's not repaired before the next round. So in E. coli, there's a two-protein system that controls this SOS system. And since you just went through all of that, all those different mechanisms for regulating gene expression in E. coli, This should hopefully make sense. There's a repressor protein. There we go with that term again. There's a repressor protein called LEX-A. All right. And it's going to bind to an operator sequence that's in front of each of the SOS genes. So there's a suite of genes that need to be turned on. They're not part of an operon in this case. But each of those genes is normally repressed. That means they're not under normal conditions. The genes are not being transcribed because the LEX-A protein is bound to that controlling element. There's another very important protein in cells called Rec-A that got its name because mutants of Rec-A have defects in DNA recombination, genetic recombination. But this protein does a lot of different jobs in the cell. Now, what happens is Rec-A has a very high affinity for single-stranded DNA. So if there's any single-stranded DNA in the cell, the Rec-A protein binds to it. Okay. Now, the binding of the Rec-A protein to the single-stranded DNA activates a protease associated with the Rec-A protein. What's a protease going to do? They degrade proteins. And what's going to be the target of the Rec-A protease? Right. If the Rec-A protein is bound to single-stranded DNA, that means there's been severe damage to the cell and there's large stretches of single-stranded DNA. That activates the protease activity of the Rec-A protein, and it degrades the LEX-A repressor protein and turns on expression of the genes involved in the SOS system. Okay. Here's the key point. This is called error-prone repair. All right. So what was the problem? The polymerase, DNA polymerase 1? What's the main one? 3. Okay. I knew that. So DNA polymerase 3 is stuck at one of these pyrimidine dimers. It can't replicate past it. What happens when one of the proteins as part of this SOS repair system replaces one of the proteins in that RNA polymerase complex and allows it to be a little more flexible. It allows it to replicate past, it lets it get sloppier. It allows the polymerase to replicate past, but in doing so, it's just going to incorporate any base to fill in the gap. So it's called error-prone repair. The cells are going to be able to save themselves by being able to replicate past that lesion in the DNA, but the price they're going to pay is a higher level of mutation. Okay? So one way I think about it, okay, everybody, remember, there's an old movie, goes way back, Butch Cassidy and the Sundance Kid. Okay, I'll set up the premise for you. So they're trapped, they're on the top of a mountain, the police are coming, and the only way is over the cliff and into the river. And one of them starts complaining because he doesn't know how to swim, and the other one says, who cares, the fall's probably going to kill you anyway. So that's what the cell's in this situation. If it doesn't replicate past that lesion, it's going to die. The consequence is it's got to take the chance. It's going to have a higher level of mutation because the polymerase is not going to be specific, but at least it has an opportunity. So that's why it's called error-prone repair. You can repair the damage, but there's a price to be paid. Okay. So to try to tie this back to what we talked about before, what type of regulatory system would this SOS repair system be? And since we only had the exam a week ago or so, you should. I'll go to a minute and 15 seconds on this one. And you thought you were done with this. I'm just helping you get ready for the final.
You never have
No, it's all cumulative.
other classes do it too.
Yes, science has that nature of all of it integrates together. All right. So what's the answer? I don't know. I don't care. All right. All right. So why is it negative? The protein is being bound. The protein bound to the DNA blocks transcription. So that's a negative system, and it's inducible because the normal system is off, and you have to induce it. You have to do something to it to turn it on. So that's a brief review for the final exam. Okay. Here's a more realistic question. One may be more typical of what you might see on the next exam.
Is it radiation?
Hmm? Radiation? That's actually the verb form.
Oh, it is?
Following UV irradiation of.
Oh, I never knew that.
Hmm? Now you got me questioning my.
I don't know if the radiation is a noun.
UV modifies the radiation. I'll go to a minute and a half on that one. Good question. You need more time? You're just saying that to keep me from moving on. But we'll go to 2 minutes on this one. Okay. Almost half of you got it right. All right. So here we go. All right. So what's the answer? All right. Okay. Any questions on this one, or should we go through it? It's just relating it back. It's just another type of gene regulation in prokaryotic cells. Okay. Just as a small point, the system is very similar in eukaryotic cells, except it has a specific DNA polymerase called ADA, and it specifically replicates past the pyrimidine dimers by putting AA opposite the dimer. So evolutionarily, there's a selection. The highest probability is going to be a TT dimer. So if the system is going to put A's across from the T's, the probability of it getting correct is higher. Okay. But it does the same thing when you have the CT dimers, so that you end up with these CG to AT transversions. So instead of being a whole repair system like we just had with SOS, this is a specific polymerase just called the error-prone polymerase. But it's doing the job of that SOS in eukaryotic cells for what the SOS repair system was doing in E. coli. Okay. I'm not going to go into this in detail, though it's interesting, and it's probably discussed in detail in the textbook. But Bruce Ames developed an assay a number of years ago, and the idea was that it was assaying for DNA mutagens. For many years, the federal government required companies that were going to put some new chemical pesticide or whatever on the market, that it had to pass the Ames assay. So the rationale being, if it was a DNA damaging agent, then the probability of it being carcinogenic was also very high. This is no longer used. There's other assays that are used now. It was based on reverse mutations. That is, you start out with a mutant phenotype, and you would be able to reverse it back to the wild type. Why would you do that? What's the spontaneous mutation frequency ballpark? Let's say we do this in E. coli, 1 in 10 to the 9th, so that's 1 in a billion. And usually the threshold was if whatever chemical you're using doubled the spontaneous mutation frequency, that was unacceptable. So if you're going to go from 1 in a billion to 2 in a billion, if you just plate cells out, I don't know, could you accurately count a billion and distinguish one? Okay. So what you're doing is you're looking for a rare event. So what he was using were oxytrophs. These were histine oxytrophs, actually from using salmonella. My guess is he had those strains already. And so you'd plate them on minimal medium. We've actually got some of these strains in the lab because I had a high school student a number of years ago used them in a science fair project. He actually went on to an international science fair with it. It was really pretty good. So the idea was, if you just take these histidine mutants and you can do this with bacteria easily, you put 1 billion of them or 10 billion on a plate of minimal medium, would you expect any to grow? Normally, if it's a histidine oxytroph, will it grow on minimal medium? But if you put several 100,000 or several 100 million on there, approaching 1 billion or so, depending on what the particular mutation was, you get 2, 3, 4, 5 colonies growing spontaneously. And that was the result of a spontaneous reverse mutation, or it could be a second site mutation, but a spontaneous mutation to a wild type phenotype. Then you would take in comparison, you would add these same cells to your chemical of interest, and if you got 10 spontaneous revertants and 20 spontaneous with your chemical, then it failed the test. Okay. But you can read about this if you want to read about this in more detail. Okay. Some other interesting experiments in exposure to humans. We all have low-dose cosmic radiation every day, unless you don't get outside. Okay. Those of us like myself who spent many, many years using low levels of radioisotopes on a day in and day out basis, the question of whether there's any long-term health effects from that, it's really very, very difficult to do those type of experiments because the level is so small. The increase above the spontaneous level would be so small. However, we can have some very acute exposures. So this is what happens in thermonuclear blasts and such. Or if your dentist decides to take a coffee break while they're doing the x-rays, those sort of things. Then you can have very acute situations all the way up to radiation sickness, and it's also lethal. So some studies of the children of parents who were exposed to these high levels of radiation between the 2 atomic bomb blasts in Japan during World War II turned out to be rather interesting results. We don't really understand all the answers. The question is, if you look at the frequency of mutations in the germ lines, in other words, what happened of those individuals who survived that exposure? And yes, many had increased incidence of cancer, radiation sickness, and such. But those who still had a very high level of exposure to this radiation, yet the question was, what about their children? What kind of mutations are occurring in the germline of these individuals? And the interesting thing was, if you do these experiments in animals, you tend to find that there is a high mutation frequency in the progeny, just as you would expect. But in this case, if you look at the children from the survivors of these atomic blasts, they really didn't have that higher level of increase of genetic mutations, different phenotypes, higher incidence of cancers and such. So for some reason, unlike what you see experimentally in animal models, in this unfortunate experiment with humans, you actually did not see the level of mutations in the germline that was expected. Don't really have a good explanation for that.
But was the reproduction rate the same?
Don't know. Probably. I mean, obviously it's going to be a limited number, but because you have to be only among the ones who survived and who still were now fertile
Yeah.
and such. It's just the interesting anomaly that you get. It's another one of those cases you get a certain result using animals, and then when you really extend this to the human situation, you get a slightly different, unexpected result. There's not a full explanation
I
for
was
it.
just thinking maybe a lot of them died before they
It could be that. It could be you can't really quantitate what the exposure level was. Based. So, just an interesting observation. Okay. So in the last few minutes, you want me to talk about something else? Please, yes, okay. All right. So in the last few minutes, I'll just start talking a little bit about transposable elements. And there's another type of interesting genetic mutations. These have been called different things: jumping genes, mobile elements, transposable elements. They all mean the same thing. They're pieces of DNA which have the ability to apparently integrate into chromosomes or plasmids in bacteria at will, wherever they want. Most of them don't seem to have any specificity as to where they're inserting into the DNA. They're a lot like viruses that can create these proviruses, except they can't replicate. They can't get out of the cell. All right? I don't think we had it on there. They were first identified by Barbara McClintock back in the late 1940s. She came up in that chapter we talked about chromosome rearrangements. And she was a maize cytologist, probably the best corn or maize geneticist in the world at that time. Just about the time that all those landmark experiments on DNA were being done, and the scientific community was finally accepting that the genetic material was encoded in DNA, this woman comes along and says, oh, by the way, the DNA is moving around. And much like Mendel's work, nobody understood what McClintock was talking about because they didn't have the right context to understand it at the time. So her work, I see a lot of parallels to Mendel's work, was pretty much dismissed for about 30 years until these jumping genes, which is what she was looking at, were in fact found in E. coli. Now they've been found in every organism that's been investigated, including humans. She did win the Nobel Prize for her discovery, but it was about 40 years after she did the work. We talked about how the human genome has all this formerly called junk DNA, now the dark matter of the human genome. But all these repetitive sequences that are so prevalent in the genome, probably about half of that is in fact the result of transposable elements. The question is, why are there transposable elements? What's the real function of them? They are a source of mutation. They can be highly disruptive. Okay, if this, let's say, this transposable element's 10 kilobases, and it inserts itself into the open reading frame of a gene, will that probably affect the function of the gene? Yeah, I sort of think about it this way: you've got some old VCR, VCR. Go to a museum, okay? There's a thing called VCRs. All these things, all these examples I used to use now, you guys don't even know what I'm talking about, okay? So you splice in, you know what it means, okay? I'm being sarcastic. It's what I do, okay? Well, let's just say you're watching some VCR. You got a VCR tape, and you're watching, Disney's Snow White, and right in the middle of it, somebody spliced in about 10 minutes of the Terminator. It tends to disrupt the continuity of the gene, just as it disrupted the continuity of the film. So these introns can insert in and jump out, and some people have called them parasitic DNA. Others say they are a source of mutations for evolution. So that question is still debated. But they're out there. They are ubiquitous. As I said, to my knowledge, no organism that's been investigated has been found to be free of these transposable elements. Because of their homologies, you can get DNA rearrangements, transversions, so they can cause a lot of havoc to the genetic material. All right. So let me just take you through the next, just hang with me because this will be a good stopping point. We'll come back and look at this because now it gets a little confusing. When you look at the structures, okay, wait a minute, another point that you don't have to tell all of your friends who are at the beach today. There is a lot of material on transposable elements in this chapter, all right? As I remember it, it'd probably take 2 or 3 lectures to go through all of it. I'm just hitting a few high points. So use what we're talking about here in class in terms of what I'm going to expect you to know for that 4th exam. So there is a lot of material in there we are not going to cover. Okay? The interesting thing about these transposable elements is when you look at the ends, when you look at the termini, what you find are little short direct repeats. Direct repeats mean it's the same sequence on the same strand going in the same orientation, not opposite strands. Inverted repeats are on opposite strands. Okay? The direct repeats are flanked by inverted repeats. Okay, confusing enough? All right, just stay with me for a couple of minutes, and then we'll come back and do this on Monday. There's an enzyme called a transposase, and it's going to recognize these inverted repeats. So they're necessary for this mobilization of the transposon. Here's the key point I want to show, and then we'll come back and talk about how this functions on Monday. The inverted repeats right here and here, those are the physical ends of the transposon. The direct repeats that flank it right here in this darker color and over here are derived from the chromosomal site where the transposon inserts. It's part of the mechanism of how it inserts. So hang with me for one more slide because then it at least puts it in your head. You can forget about it till Monday and we'll come back to it. Okay? What happens here is as the transposon is inserting, there are staggered cuts in the DNA. Instead of breaking the DNA at the same place on both sides, it's cut right here where these 2 arrows are. The only thing holding these ends together, it's like similar to a restriction enzyme, the only thing holding these bases together is the hydrogen bonding. The transposon inserts, but now we have these gaps. Those gaps have to get filled in by the DNA repair system, and that's how the direct repeats are created. So they are the result of the chromosomal sequence. We'll start back on this again on Monday.